Hello all. So welcome to yet another video of amino acids. Thank you for your support in my previous video. I hope you support this video too and you will enjoy this video more. So today we are going to discuss the net charge. We are going to calculate the net charge on any peptide at any particular pH. So on the board today I have mentioned the pKa values of different groups that can be ionized at any particular pH. Okay. So when we talk about NH3 group, it is ionized at 9.6 pH. When we talk about the main chain COOH group, it will ionize at 2.4 pH. When we talk about the charged amino acids, the amino acids that have ionizable side chains. We have talked about amino acids that have non-ionizable side chains and the amino acids with ionizable side chains. So the amino acids with ionizable side chains have certain pKa values at which their side chains are ionized, right? So for histidine, it is 6. For lysine, it is 10. For arginine, it is 12. तो इसको आप ऐसे you can just learn it like this, that HRK 6, 12, 10. So just say it 5 times, you will be able to learn it. HRK 6, 12, 10. 6, 12, 10. Alright. So we have another ionizable groups containing amino acids, the acidic amino acids that have acidic side chains. That is, they have COH in their side chain. So we have aspartic acid. Aspartic acid side chain will ionize at 3.6 and for this it will ionize at 4.25. Alright. So these were the pKa values for certain amino acids and the main chain amino group and the carboxyl group. Now we'll discuss certain examples and we'll see the net charge on any given peptide at any particular pH. Alright. So let's begin with today's topic. Let me discuss what is main chain amino and carboxyl group. That will be much more clear to you. Suppose there is a peptide. Always when a peptide is ribosomically synthesized, its NH3 terminal is on this side. The first terminal, the first amino acid will have the free NH3 group or the amino group. The peptide group is always read from amino terminal to the carboxyl terminal. So first we will write the amino terminal of the peptide. Then we will have the amino acids. For example, there is A1, A2, A3. I hope you are clear that these are amino acids, and A4. Then we have the carboxyl terminal. So we have the amino terminal and the carboxyl terminal, and the main chain pKa values that I'm talking about, it is of these groups. This NH3 values pKa is 9.6 and the COH pKa group value is 2.4. Are we all right? Are we clear with that? So now we will discuss and calculate the net charge on any peptide at any given pH. Okay, let's just take an example. We can take any name. All right, we can take any name. Let's take an example of Maharashtra. Maharashtra. Of course. So we can see, so I'll give you a question. So you have to solve it for me. So I'm saying that on pH 10, what will be the net charge on this peptide? Calculate it for me and give the answer. So note it down. On Maharashtra, what will be the net charge on the peptide at pH 10? I hope you have taken the screenshot and noted it down and solved it on your own. Let's solve it now. Okay. So we have first amino acid as methionine. So have I told that methionine has an ionizable charge group on the side chain? No. Alanine has any ionizable group? No. Histidine does have ionizable group. Yes, right? It does have ionizable group. It is a basic amino acid, so it will have NH3 as its side chain in the side chain, and it is an ionizable group. The amino group in its side chain, which is an ionizable group. It is a basic amino acid. Alanine, I told you that it does not have any ionizable group. Then R, that is arginine. Arginine is the most basic amino acid and its pH value, the pKa value of the side chain lies in the range of 12. So you can see how much basic it is, that on increasing that much pH to pH 12, then only it loses its H plus ion. Alright. So for the R, we have to take that yes, it has an ionizable side chain. A does not have any ionizable side chain. Serine does not have any ionizable side chain. Histidine, we have just discussed that it has ionizable side chain. Then T does not have any ionizable side chain. R does have an ionizable side chain. A doesn't have, and we have to of course calculate for these two as well. Alright. So by pH 10, we have to see what will be the charge on this peptide. So at pH 10, we can see that the pKa value of NH3 group is 9.6. So by 9.6, this will become NH3 positive, say this will become NH2. So earlier the peptide had +1 charge. Now it will have, because of the pH 10, it will have net 0 charge on the NH3 group. Now we are going to talk about the histidine. Histidine also had one NH3 positive group in its side chain. Now it will also ionize by the pH 6. As I have told you earlier that for histidine the pKa value of the side chain is 6. So 6 pet will get ionized, but now we are taking in consideration the pH 10. So at pH 10, of course, histidine will also be ionized and the charge will be 0, because if NH3 will give its H plus ion, it will become NH2. So arginine has basic side chain. It has NH3 positive group in its side chain. So we can say that it will remain like this because its pKa value is 12. All right. Now we're going to talk about histidine. It will remain same at pH 10. The charge on the side chain will become 0. For R, the side chain will have positive of 1 charge. COH minus COH group will convert into CO minus group. It will convert into CO minus group. Why? Because at 2.4, its pKa value is 2.4. So by 2.4, it will get ionized and the COH group will convert into the CO minus group. So now we will calculate the net charge on this peptide. So NH3 ka, of course, it will be 0. Histidine we have calculated at pKa for pKa 6, at pH 10 it will be 0. Arginine it will be, of course, +1 because pKa was 12. For histidine again it will be 0. For arginine at pKa 12 and at pH 10 it will be +1. So plus and minus 1 will be cancelled. So the net charge on this peptide at this particular pH will be +1. Are we clear? I hope all your answers are correct. All of yours answers are correct and you have got +1 as the net charge on this peptide at pH 10. All right, good. Chalo, let's take one more example so that it will be much more clear for you people. Let me take my name in this one this time. And maybe my brother's name too. Okay. So we have taken the two names and not two names. Now, in this case, it's a peptide now. So we have a free amino terminal, of course. And we have a carboxyl terminal at the C terminal at this end of the peptide, right? So now you're going to tell me that which are the ionizable groups, which are the amino acids that have ionizable side chains. So time begins for you people. You will take just 5 seconds and mark all the amino acids that have ionizable side chains. So time begins now for you people. I hope you guys have marked. Let me mark for you now. So first of all, D. D is what? D is aspartic acid. Aspartic acid is an acidic acid. Acidic acid means it is having an acidic side chain or the ionizable side chain. It has COOH in its side chain. It has COOH in its side chain. So it can be ionized into COO minus group. So yes, we can take as D that D is containing an ionizable side chain. Then I is isoleucine. It does not have. Y is tyrosine. No, we didn't talk about that. Alanine, no. H, histidine, yes, we did discuss in the last question also. A, no. R, yes. S, serine, no. Histidine, yes again. And COH, yet again, we got one ionizable group in the peptide. All right. So NH3 positive group. So we can say, suppose if this time I take pH, let's take, what pH do you want me to take? I think we can take pH 6 this time. Okay. So at pH 6, NH3 positive will be what? It will be ionized because it will be ionized. It will be ionized because at pKa NH3 positive group, for NH3 positive group, the pKa value is 9.6. At pH close to 9.6, only then it will leave this H plus ion and become NH2. But right now in the question, we have taken pH 6. So for pH 6, the charge on this peptide ka NH3 group, the main chain NH3 positive group will be +1. All right. Now for the D, for the aspartic acid, as we have studied for the aspartic acid, the pKa value was what? It was 3.6. So for 3.6, the pH given is 6. So this COH will convert into COO minus. So the charge on this group will be -1. So we have +1 here We have -1 here. Now we'll talk about histidine. Histidine loses its H plus at pH number 6. pH 6, it loses its H plus. Okay. So for histidine, it will be 0, because what will it become? It will become NH3 positive to what? NH2 group. All right. So it will have a net 0 charge. Now for arginine, arginine loses its H plus at pH 12. I told you it's a very, very basic amino acid. So it for this will be +1. And for histidine, yet again it has come, so it will be 0. For COH group, it is 2.4. All right. So pH at 6, it will be -1. So what will be the net charge on the peptide at pH 6? Cancel -1 plus 1, -1 plus 1. The net charge on this peptide at pH 6 will be 0. I hope you guys calculated it before me solving it, and that will be very helpful for you people. And these kind of questions do come in the exam. They'll give you certain peptide with maybe their 3-letter abbreviation or 1-letter abbreviation, and they'll ask you that, what is the net charge on this peptide at this particular pH. Are we clear with this thing? Let's continue with the next topic. And you can do more questions. You can just write the names of maybe like your name or your friend's name, and then just maybe calculate the charge on that peptide. Okay. Thank you. Let's begin with the next topic. So now we are going to discuss about the titration curves of amino acids. So I have told you already that amino acids have two ionizable groups, that is your NH3 positive group and your COH group. So because of these two groups, the titration curve for the amino acids we'll be plotting, and we'll be checking at what pKa values these amino acids will lose their H plus ions. Okay. So what we're going to do is we're going to add the OH equivalents. So for example, for OH equivalents, we'll add NaOH in the solution, and we'll check that pH will increase. Of course, we'll increase the NaOH in the solution, the pH of the solution will increase. Okay. But then we will also add our analyte, and we'll see that at what pH values it will actually neutralize the H plus, the OH minus ions that are there in the solution. Okay. So we have added the NaOH in the solution, and we will add our analyte, and we'll check the pH values. Okay. Fine. So this titration graph I am plotting for which amino acid? The most basic amino acid, not the basic amino acid, like the most basic amino acid, that is glycine, jiske side chain mein there is no group, there is only just H ion. So there are just two ionizable group in the structure of glycine. Let me draw it for you, the structure of glycine. So we have one COH group, of course, we have the NH3 moiety, we have the H plus group, H group, and we have the another H group in the structure. So we will see now, and I have told you that the pKa values of NH3 group, NH3 positive group of the main chain is 9.6, and for COH it is 2.4. Yes, I told you already, right? So now we're going to draw the titration plot of this amino acid glycine. So as we increase the NaOH in the solution, the pH value of this solution will increase. But now what happens is this COH group will sense this OH minus and will start releasing the H plus ions, right? And it will become ionized. It will become COO minus group. So what will happen? It will try to release the H plus ions in the solutions, and finally when all the COH group have released, they have become totally COO minus. So now what will be the structure of our amino acids? Let me draw it for you. Now I have told you that the COH group has been ionized at this pH around 2.4. So what we can draw is now our amino acid has become what? COO minus and NH3 positive, and these will remain the same. So our COO group has been totally ionized. It has become COO minus group. Now we'll draw the same for the NH3 group. Now what will happen as we keep increasing the pH gradually? What will happen? The NH3 will also start losing its H plus ions and will tend to become NH2 group, right? So what will happen? It will try to become NH2 group. So the second peak that we'll get here. So this peak is for COH group. This peak is for NH3 positive group. Now this NH3 positive group will become what? It will become NH2 group. This will become what? This will become NH2 group, right? So this is the pKa 1. which will come around what pH? It will come around 2.4. Now this is pKa2, which will come around which pH? That is 9.6. So we have drawn the titration curve of amino acid, that is glycine. So first pKa1 is 9.4, the other one is 9.6. And remember this thing, pKa is the value at which half of the molecule is ionized. That means at pH 2.4, half is in CO minus form, half is in COOH form. Then after that, again, as the pH increases, it will actually convert whole into COO minus, right? Now there will be a point when there will be what? There will be an isoelectric point that will come in midway, that there will be a formation of zwitterion. What is the zwitterion? Zwitterion is that molecule for which the net charge on the molecule is 0. That is called the zwitterion, and the point at which, the pH at which the net charge on any molecule is 0 is known as the isoelectric point. It is also designated as Pi, and it is calculated as pKa1 plus pKa2 by 2. Are we clear? So it will be pKa plus pKab, matlab pKa for the side chain COH, for the main chain COH, for the main chain amino divided by 2. So that will be the isoelectric point for the glycine. So at that pH, we will have the formation of zwitterion in the solution. So what we are going to write for the Pi value? So for the Pi value, what we are going to write? That is 2.4 plus 9.6 divided by 2. So 2.4 plus 9.6 will be, calculate, it will be 12. 12 by 2 will be 6. So Pi of glycine will lie somewhere here, where there will be a formation of zwitterion. Now what is zwitterion? Zwitterion is this. COO minus and NH3 positive group is there. So now the net charge on this peptide is 0, because this is minus 1, this is plus 1. At this point here, at pH 6, what we can say? That the glycine amino acid is forming a zwitterion. It is having the isoelectric point. At what pH? At pH 6. So now we have discussed about the glycine. Now we will discuss about one acidic amino acid and one basic amino acid. Then you will be more clear with the titration curves and the pK values of the amino acids. Thank you for watching my video. In the next video, we will discuss the titration curves of glutamic acid or maybe aspartic acid. Maybe we'll discuss about aspartic acid because it is the most acidic amino acid, and I'll also tell you on the criteria on basis of which it loses its H plus ion, and we'll discuss about one most important basic amino acid, and you will tell me in the chat section that which amino acid is that which can act buffer at the physiological pH. I hope you will answer in the chat section, and we'll discuss about that titration curve in the next video. And in the next video, I'll also discuss the tricks to learn the amino acid structure and significance of each and every amino acid in our biological system, and what alterations or what modifications we can make to these amino acids. Thank you so much for watching this video. Thank you for giving support in the previous video. I hope you have enjoyed this video more and you will support this more. Thank you so much. Thank you.